Oh, did you get the, about 115? Were you able to access that?
I didn't take a look yet, but I can check right now if you want.
You want to check it? Don't play it for everybody to hear.
Okay.
We don't have that facility in this class.
But thank you for posting it.
That's a high tech room.
Yeah.
This is low tech.
6 weeks left.
I got a high-tech room too. I mean, we're trying. It's no lecture capture, but it'll do.
They're now a gene that people are born with these things in their hands, so it seems that's the case.
I think so. My youngest brother got his first phone when he was, I want to say my mom bought it when he was 8, but he was riding the bus home, and she wanted to make sure that she could get in touch with him at all times when he was walking onto the street. She'd be at home, but she wanted him to have that little bit of independence so that he could walk in. So that's what she did. But he is 17-gallon drives.
Thank you.
All right, I have 3 exams left for people who didn't pick it up on Tuesday. So if you weren't here Tuesday, you want to pick up your exam, do so after the class. Sorry. Okay, so we're going to talk about cell signaling today. Remember, all events that we described this semester really involve cell signaling. You have receptors which bind to a ligand, and then there's some kind of change that takes place metabolically, physiologically in that cell after receptor and ligand dock with each other. So we could do the whole course talking about cell signaling, but I've restricted it only to T cells, and we're going to talk a little bit about TLRs at the end of the discussion today, if I get there, that is. And now, a course taught by my colleague, Dr. Cagle, in cell biology involves a lot of cell signaling. That's what most of that course entails. So if you're interested in cell signaling, I advise you to take Bio 46 if you haven't done so already. But if you're graduating senior, you can take it next semester. It's still one more semester left to you. How many people here are graduating seniors? Well, you have the opportunity to take a course in cell signaling, not entirely, but a lot of it is. So we're obviously not going into detail that Dr. Cagle goes into in her course because she spends weeks on it. We're only going to spend a day or so on it. But we'll get the highlights and what actually happens during cell signaling. And in this case, we're going to look at the TLR when it encounters an APC, for example. And there are 3 phases of activation which can take place. And one, most of them are transcription factors, which happen pretty quickly that we activate transcription factors. And that's what we're going to limit our conversation to today. We're only going to talk about transcription factors and how they get activated after TCR engages the correct ligand. You can see there's 5 of them here. We're going to talk about 4 of them. CFOS, CJUN, nuclear factor activation of T cells, of activated T cells. And this is NF-kappa B, nuclear factor kappa B. By its name, you may infer that this particular transcription factor was first identified in B cells, hence kappa B, with the expression of kappa light chains. So we're going to skip C Mike. So these are the 4 things we're going to talk about. But after transcription factors are activated, the cascade takes place, and we get a whole bunch of cytokines which are transcriptionally then activated in our T cell, depending upon what T cell type we're talking about. We're not going to talk about the activation of each of these cytokines, in fact, none of them. That's just a downstream effect of having the transcription factors become activated. And finally, there are late molecules which are regulated later. Class 2, remember, is not really shown on T cells regularly, so you skip this in your mind because I don't want to get you confused. So we'll cross that out. There are exceptions, as I say in immunology all the time, and this is another one, but let's just cross that out for your class because You don't want to get confused. T cells normally do not express class 2, but they certainly do, after they become activated, express adhesion molecules, and that's something to allow the T cell to engage its target with a higher affinity. All right, but again, we're going to limit our conversation, time being what it is, to transcription factors. Now, this is just a quick review of very simple biochemistry of the cell membrane. And as you know, I hope, there are two leaflets of a cell membrane, and this is the inner leaflet. That is the leaflet which is in contact with the cytoplasm rather than the extracellular fluid. Here we are. And now, as you well know, I assume also, most of the lipids in a cell membrane are made up of a glycerol backbone, okay, shown here, and esterified to two of the hydroxyl groups are fatty acids, shown up here with various sorts of them. And in the third position, hydroxyl group, we have a phosphodiester bond of an alcohol in this case, which is called inositol. Okay, so here is phosphatidylinositol. So phosphatidylinositol is the key molecule that we are going to worry about and how we're going to activate it. Again, just to get your geography correct, it's located on the cytoplasmic side of the cell membrane. And again, here's the glycerol backbone, and here is the two fatty acids. Now, phosphatidylinositol can be converted to have one more phosphate by an enzyme called phosphatidylinositol 4 kinase. And for heaven's sake, you don't have to memorize these enzymes at all. And that gives you another phosphate group over here in the fourth position. And then there's a second kinase, which is phosphatidylinositol P5 kinase, which adds yet another phosphate group. So we have 4 and 5. And this is the molecule we're going to be talking about today in somewhat detail, PI45 bisphosphate. This is the molecule we're going to talk about, and how we can convert it biochemically into two other molecules which are involved in cell signaling. Now, to make a long story short, our T cell activation is going to activate an enzyme, and I do want you to know this enzyme, called phospholipase C. And you just have to memorize it. And of course, the abbreviation for that would be PLC. And phospholipase C works on PI45 bisphosphate, or PIP2, to give you two products. One product is the inositol, which we now have 3 phosphates on it. Okay. Here's the third one, and these 4 and 5. So that's inositol 1,4,5 trisphosphate. That's going to be a very important signaling molecule. And the second molecule is this rump molecule left behind. We have glycerol, and the third carbon we have hydroxyl group, and then the 2 fatty acids remain. And this molecule, because it has 2 fatty acids, we call it diacyl, 2 fatty acids. And the backbone molecule is glycerol. So this is 1, 2, diacyl, meaning first carbon and second carbon have fatty acids associated with it, esterified to it, and it's a glycerol backbone. So diacylglycerol. So those are the 2 molecule names we have to know to understand cell signaling in terms of the T cell. So inositol 1,4,5 trisphosphate, which I'm going to call IP3 from here on in, just for convenience, my convenience. IP3. And DAG, I'm just going to call this DAG, 1,2-diacyl. But what I want you to keep in mind is the parent molecule, PIP2, and this enzyme, phospholipase C, which gives us the 2 products which are important, diacylglycerol and IP3. Okay, so keep that in mind, and then we're going to learn how we can activate and what these molecules do. So we'll learn how to get these 2 molecules and what those 2 molecules do. And one of our first choice here is, first of all, how to figure out how we're going to activate phospholipase C. So our story's really going to start with figuring out how we're going to activate phospholipase C. All right, so that's the background information, general information. And now we're going to talk about what happens when the T cell engages its target. This is just an overview of parts of the T cell receptor you already know. So here, we're not talking about gamma delta T cells, although operationally they're very similar. We have alpha beta T cell receptor, we just finished discussing last time. And here are CD3 molecules and the zeta zeta. And I already told you that some people consider zeta zeta part of CD3, some people consider it to be distinct. It doesn't matter. It's splitting hairs at this point. And the CD3 molecules per se, gamma epsilon and epsilon delta, have ITAMs. Here's what the acronym stands for. But the most important letter to remember from this acronym is T for tyrosine. Because when these tyrosines become phosphorylated, they can then lead to activation of cell signaling. On the other hand, when these tyrosines become dephosphorylated, that shuts off cell signaling. So the on-off switch really relies upon whether tyrosines are phosphorylated or dephosphorylated. Now, the signaling component, which has the most ITAMs, and hence the most tyrosines, are the zeta-zeta homodimer. And you can see they have 3 ITAMs as opposed to 1 for gamma epsilon and epsilon delta. So our story today won't really talk about gamma epsilon, epsilon delta. I'm going to confine it to the zeta-zeta signaling cascade. These same processes are also occurring in gamma epsilon, epsilon delta, but they're less powerful for obvious reasons, because there's more tyrosines in the zeta-zetas. All right. So our goal is to activate phospholipase C, but before we can do that, we have to phosphorylate the tyrosines on the ITAMs of zeta-zeta. That's our mission. Everybody get what we have to do? And once we get the tyrosines phosphorylated on these ITAMs, then we eventually can lead to activation of phospholipase C, which then can work on PIP2 and give us diacylglycerol and IP3. Now, here's a cartoon of what's probably going on in our T cell. So here we have the T cell receptor. Here's the APC. All information you're very familiar with, I know at this point. And here's the class 2 molecule, the processed peptide. So we have the right restriction and the right specificity. We have these CD3 molecules and our zeta-zeta associated with it. And also, this is a CD4 cell, so we have our CD4 molecule. A theme which is repeated almost always in cell signaling is dimerization. So you have a single TCR engaging the APC, but that leads to a dimerization or a connection to at least 2 different T cell receptors, cross-linking of them, in this case the CD4s. Here's the APC, the CD4s line up. They actually lead to aggregation to T cell receptors. When that happens, that brings about the phosphorylation, we're going to learn how, of the tyrosines on our zeta-zeta. The little red balls means they've been phosphorylated. And that, once the tyrosines become phosphorylated, here's another very important key step. Once these tyrosines become phosphorylated, they act as docking sites for one of the key molecules, key control molecules in T cell signaling. This key molecule has a molecular weight of 70 kilodaltons, and it's a zeta-associated protein. Zeta-associated protein. It's called ZAP70 from here on in. ZAP70. And ZAP70 is this little bean-like structure here with a little hat on it. and this little bean-like molecule can dock on an ITAM, but only if the ITAM is phosphorylated. So this little binding site is called SH2, and that can bind to phosphorylated tyrosines on the ITAMs. All right, so let's start our story again. We have the T cell receptor hooking up with the antigen-presenting cell. That causes dimerization. Our first goal is to phosphorylate the tyrosines on the zeta-zeta ITAMs. So that must mean we have some kind of kinase which can phosphorylate the tyrosines on ITAM once we get that cross-linking and engagement with the APC. Not too surprisingly, as I said, those molecules are called kinases. And the kinases which are involved in the initial activation of T cell signaling are called SRC kinases, so called because they are homologous to the type of kinases we can find in the SARC virus, sarcoma, but that's not our concern here. All right. Now, in cell membranes, we often have a structure called a lipid raft. A lipid raft has less fluidity. A lipid raft tends to be more stable than other parts of a cell membrane. And the reason a lipid raft is more stable than other parts of a cell membrane, because instead of having those phospholipids I showed you, which have mostly, again, glycerol, 2 fatty acids, and something else is sterified to the third hydroxyl group, or phosphodiester bond. We have instead something called a ceramide, or sphingolipid. And the sphingolipid consists of a fatty acid linked by an amino group to sphingosine. Sphingosine has this amino group to which a fatty acid can link. And we have an R group there, and we have various things in the R group. It's just hydrogen, and it's ceramide. If it's phosphocholine, it's sphingomyelin, etc. But interspersed with our phospholipids, we have these regions of our cell membranes which have rich amounts of sphingosine components. And again, these parts of the cell membrane are more rigid, and more likely to stabilize all these complex molecules involved in cell signaling. Because you don't want the membrane to be highly fluid. If that were the case, all these various components could drift apart from each other, and you wouldn't get efficient cell signaling. So you want to stabilize this interaction in a part of the membrane which has less fluidity. And when we get cross-linking, this is showing you a B cell receptor, but the same thing would be true for a T cell receptor. It's the only figure I can show from your book. But in this case, when the T cell receptor, or in this case a B cell receptor, gets cross-linking, that tends to drag the TCR, or in this case the BCRs, into the lipid raft and into an area which has high amounts of kinases. These are kinases shown here. So let's review again. So we have the TCR engaging an APC. And that gives us cross-linking for at least 2 different T cell receptors, undoubtedly more than just 2. That tends to draw these components into a part of our cell membrane, the T cell membrane, which is rich in a sphingosine compound, which has more rigid structure than the phospholipid. And in that lipid raft, we also have nestled or embedded, if you will, kinases. And the kinases we want to talk about in the T cell are called SRC kinases. All right? So our first job here is to get cross-linking. And then the SRC kinases normally are inactive themselves. So there's a lot of steps we have to go through to get cell activation, which should ring in your heads why are there so many steps leading to cell activation? Why not make the threshold very low? And the answer should be obvious to yourself. You don't want promiscuous activation of T cells. You're going to, in that case, get a lot of autoimmunity if that were to happen. You want the threshold to be high enough where you're not going to get unneeded activation of T cells, but not so high that you don't get the T cells become activated when it's actually needed. So you have to have that nice medium, if you will, of activation. And the Src kinases themselves, let's just look at the right side of this. I don't know why I even have the left side. Just look at the right side of this particular figure. Should have gotten rid of the left side. Are normally inactive because they have a phosphorylated tyrosine. However, there's another molecule called CD45, which exists in T cells and in an altered form in B cells. Both cells, this molecule exists, CD45. In B cells, it's slightly different. It's more glycosylated in B cells, but that's not something we have to worry about. CD45 is a phosphatase. And when we get, again, this cross-linking of the T cell receptor with the APC, and now we're bringing all these molecules together, or Src kinases in the lipid raft, we're also now dragging in CD45. And CD45 dephosphorylates the Src kinase, and then converts it to an active enzyme. Right? So let's go through the steps that we've gone through so far. We first have to have cross-linking. That brings us to the lipid raft. That allows us to congregate with a bunch of Src kinases, which in turn are now juxtaposed to our phosphatase CD45, which dephosphorylates the Src kinase and now makes it active. And once the Src kinase is active, then it can start phosphorylating the tyrosines on the zeta-zeta ITAM. So that's our next step. So CD45 dephosphorylates the Src kinase. That causes them to be active. And the Src kinases are tyrosine kinases, so they phosphorylate proteins at tyrosine residues. And therefore, our target, of course, are the ITAMs of zeta-zeta, and they become phosphorylated. And now, how many people remember what I said about maybe 7 minutes ago? Once tyrosines are phosphorylated on our ITAMs, that creates a docking site for what important regulatory master switch molecule? ZAP70. And remember why it's called that. So let's review that again. It's really important. If you fall behind here, everything else is going to be just spaghetti overcooked. And there's nothing worse than spaghetti overcooked. It turns into mush. That's some biochemical nightmare. So we first get TCR engaging an APC. We get cross-linking of at least 2 TCRs, minimum of 2. That drags everything, it seems, into the lipid raft, which is a more stable component of our cell membrane. I know I'm repeating a lot, but I want to make sure you get all this. And then in the lipid raft, we are now juxtaposed or brought close to our Src kinase, which also now is brought close to the phosphatase CD45, which dephosphorylates the Src kinase, so it now can phosphorylate the tyrosine residues, the ITAMs on ZAP70. That's it. And now once that happens, we have a docking site for ZAP70. Boy, we're almost done. Everyone follow? Yeah. And by the way, if you're not following, I gave you a master slide here at the end, which combines all this together. And I'm sure by the time we're finished, you'll follow this master slide pretty well. Well, this Western blot shows you that ZAP70 in inactivated cells from the thymocytes or from lymph node T cells inactivated. There's a Western blot which only will recognize tyrosines which are phosphorylated. You can see ZAP70 in the resting cell is not phosphorylated, whereas in the activated cell, ZAP70 becomes phosphorylated. This is how they learned all these steps, by doing simple, well, not so simple sometimes, Western blots. And to do these Western blots, they had antibody which only reacted with tyrosine, which was phosphorylated. So we had a very specific antibody. They had a very specific antibody which recognized tyrosines and bound to it only if that tyrosine was phosphorylated. So that's what we're showing by this Western. In activated thymocytes or activated lymph node T cells, we do have phosphorylated tyrosine, shown by these two bands. So it works out pretty well, this little technique. Those who are observant notice that even in resting versus activated cells, we seem to have the tyrosines at the ITAMs on the zeta chain, whether or not we're inactive or activated, phosphorylated, which seems to go counter to our central hypothesis here. And this thing was published way back in '96, when most of you probably weren't even born. So at that time, it turns out this probably isn't correct. That indeed this data set has been shown not probably to be correct, and in inactive cells, zeta ITAMs are not phosphorylated, where in the active state they are. Although even here you can see there's more phosphorylation in the active state than there is in the inactive state. Okay. All right. So now the next step is we have ZAP70 hooked up to the tyrosines, which are phosphorylated, or ITAMs, and our next step is as follows. I guess I didn't show you that. All right, let's go back a slide. Once the ZAP70 is docked onto the ITAMs, then the ZAP70 itself becomes phosphorylated by the same Src kinases which phosphorylated the ITAMs. Okay, one of them is called LCK, another one's FIN. Both are involved. So FIN and LCK phosphorylate first the ITAMs on the tyrosines on the ITAM. ZAP70 docks Src kinases, LYN, LCK rather, and FYN. Then phosphorylates ZAP70. ZAP70 becomes an active tyrosine kinase only when itself is first phosphorylated. So we have here a flow chart of tyrosine kinases. The first ones involved are the Src kinases, FIN and LCK. They phosphorylate the ITAMs. That creates a docking site for ZAP70. ZAP70 in turn then also becomes phosphorylated by the LCK and FYN, the Src kinases. And it's at this point, when ZAP70 becomes phosphorylated, it turns into an active tyrosine kinase. This is our second tyrosine kinase. First were the Src kinases. Now the second one is ZAP70. Then it's the master switch. What does ZAP70 do? ZAP70 in turn phosphorylates its tyrosine residues to proteins called LAT and SLP76. Once LAT and SLP76 are phosphorylated by what? ZAP70, which only was active once it became phosphorylated by a Src kinase. And then once ZAP70 is phosphorylated, remember, it also becomes a active tyrosine kinase, and it phosphorylates LAT and SLP76. And then LAT and SLP76, once they're phosphorylated, can be joined together by an adaptive protein called GADS. It recognizes the tyrosine, which is phosphorylated over here. And that makes a scaffold protein. It looks like a basket in this cartoon. Not really, of course, but a cartoonist certainly does. And into that basket will slip Okay? Not a lacrosse ball, but instead, phospholipase C gamma. Phospholipase C gamma. Now, to quickly review, what is the substrate for phospholipase C? Or PIP2. PIP2. And phospholipase C, when it interacts with PIP2, gives you two products, diacylglycerol and IP3. So remind yourselves of that. This is, remember, I wanted us to get to phospholipase C. So now we're at PLC, which is activated. Okay, PLC, in order to become activated, has to be phosphorylated by another tyrosine kinase called a TEC kinase, ITK or TEC kinase. And where the heck we get this kinase from? That may be by CD28 activation pathway. Remember, that's our co-receptor. CD28 binds to CD80 or 86. That may activate a different set of tyrosine kinases, one of which are the TEC kinases, which then phosphorylate phospholipase C. Whew. All right. Now, what do we do with phospholipase C? You know the next step. We're going to get diacylglycerol and IP3. So let's recapitulate one more time. I know I'm risking being boring today, and I'm happy to do that. I want to make sure you get all these steps down. I'm doing your homework for you, because if you don't follow all these steps, then it's just a big mishmash of names. So TCR hooks up with its APC. First step after that is cross-linking. Okay. Then we, what?
At least two.
Right, by at least two. Right? Probably more. Right? And then we move into what part of the membrane? Lipid raft, which is more or less stable than our phospholipid portions of the membrane, more, less fluid, I should say. And in that less fluid part of the cell membrane, what type of molecules reside? Are SARC kinases, as well as a phosphatase called CD45. So everything's dragged together. And now everything is close enough where they can actually have substrate working with its enzyme. So now we dephosphorylate our SARC kinases. Now they're active, and then what do they do, class, together? What do they do? The SARC kinases then phosphorylate tyrosines on the zeta-zeta. At what parts of the zeta-zeta cytoplasmic tail? The ITAMs. A stands for activation, by the way, because there's another side of this coin. There's also ITIMs, which are inhibitory. So we're going to learn about those, I'm hopeful, today. So now we're still in the A, the activation part. So now we have ITAMs, because by the way, if you activate something, you have to deactivate it. So now we have ITAMs. Okay, they get phosphorylated. That causes a docking site for what master switch molecule at this point?
70.
70, which has a little hat on it, which is called SH2, and it can recognize phosphorylated or dephosphorylated tyrosines. Phosphorylated tyrosines. And then what happens when our little ZAP70 docks onto the tyrosines, which are phosphorylated on our ITMs of our zeta-zeta chain? It then gets phosphorylated by which enzymes? SARC kinases, LCKFYN. It now becomes an active molecule. And what's the target for ZAP70? Right. Right there, LAP and SLP76. And then they get joined together to form a basket. And what kind of molecule gets shoved into that basket? PLC. And PLC probably does not get phosphorylated by ZAP70, but by a different set of enzymes called TEC kinases, because nature never is direct and always tries to make things as confusing as possible for people studying this for the first time. So now, and by the way, most scientists do if it were too simple. So now we have phosphorylated phospholipase C, and this is the very first thing I told you about at the very beginning of today's class. What is the target for PLC? Phosphatidylinositol, right? PIP2, and then PIP2 gets enzymatically cleaved, and what are the two products of that? Diacylglycerol and IP. So now the next step is to tell you what do diacylglycerol do and what do IP3 do. Ta-da! All right, so that's where we are. However, your colleague, my colleague, your instructor, if you've had her, Dr. Susan Cagle, this is from her part, I think, from her thesis. I think, maybe not. I think it's in her thesis. She did some work on our story of today with T cell activation, and it's a brilliant piece of work. Even though it's from 2010, it's still a brilliant, it was then a brilliant piece of work, it still is a brilliant piece of work for time immemorial. And what they discovered in her lab was that there's a weird ZAP70, which is called an analog, and this ZAP70 substitutes methionine at position 414 with alanine. And when that happens, this particular ZAP70 is sensitive to an inhibitory molecule, which I can't pronounce, so we'll just call it PPI. Okay? We'll just call it PPI. So this ZAP70, but not the native, not the wild type ZAP70, is sensitive to this inhibitory molecule called PPI, but not the native molecule, only this weird one with this one amino acid substitution. Okay? And then they did a whole bunch of Western blots with cells which were ZAP70, native or wild type, and ZAP70 which had the AS, this variation, which were sensitive to this particular inhibitor. And then they took their T cells in culture, and they either didn't stimulate them at all, or they stimulated them with vehicle, just means buffer medium in which the cells are grown. And the way they stimulated the cells was by using antibody against CD3 epsilon and anti-IgG. When that happens, that cross-links all the surface molecules of importance in the T cell, and that leads to activation of the T cell. So instead of having an APC, we're substituting the APC by having antibodies cross-link the T cell receptors. So that's what she did. So you have anti-CD3, and then you have an anti-IgG added to the anti-CD3, and that leads to cross-linkage. And then we get all the downstream effects because of that. All right? And then what I want you to look at is, there's a lot of stuff here, but let's just look at the one with the asterisk. In the wild type versus the mutated version of ZAP70, after cross-linking, which is activation, you notice each lane, vehicle means it was just activated, the way I told you. 5 micromolar and 10 micromolar means that they added 5 or 10 micromolar of this inhibitor. And then I want you to look at LAT. Remember what LAT is. LAT is going to help us make the basket, which is going to contain phospholipase C, which in turn is going to be phosphorylated by a TEC kinase, so we then can attack phosphatidylinositol to lead to diacylglycerol and IP3. Yeah, Alana?
What did you say the mutant was sensitive to?
This molecule, PPI. Here, you want to read it? I can't pronounce it. Okay. All right. Here it is, down here.
Where?
Right here.
Yeah, that's a long one.
Yes, it is. So you organic chemists out there, have fun. I'm not an organic chemist, so I'm just going to call it PPI. All right. In any case, they use 5 micromolar or 10 micromolar. Now I want this class now to look at this particular horizontal display where we look at phosphorylated LAT at tyrosine 132. Why, of course, you know it's tyrosine and tyrosine 132. And tell me the difference between the wild type ZAP70 and the mutated ZAP70. Go ahead. Look at that. All right. Any takers out there? What happens when we add this inhibitor to the wild type ZAP70 containing cells after we activate them? Does LAT still become tyrosine phosphorylated?
Yes.
Yes. However, what about the mutated cells as we add more inhibitor, 5 micromolar and finally 10 micromolar? What happens to the amount of tyrosines which are phosphorylated on LAT at the 132 position? It becomes what? Less. Look at that. That's pretty dramatic. It becomes less, indicating that ZAP70 is essential to lead to LAT being phosphorylated, and all the downstream effects, if you inhibit ZAP70, would therefore be inhibited. However, let's look at some other stuff here temporarily. What's the difference between the wild type and the mutated type of ZAP70? There's a difference between the wild type, these flow lanes, and the mutated ZAP70. What do you see there? Remember, you're an observer. It's the first time this data has been shown. There's something you should look at, which is interesting, which was not expected.
Bands are heavier.
What was that?
The bands are heavier for the wild type.
Right. So that tells us what? About the concentration of ZAP70 in the mutated form, there's what?
Decrease.
This is the mutated. There's
It
more.
increases, yeah.
There's more. There's more of it, right? So there's more. There's more ZAP70, which is interesting. They didn't expect that to take place. There's more ZAP70 actually in the mutated T cells than there is in the wild type. And by the way, when you do research, sometimes you're going to get data that you don't expect, and then you have to interpret it. It's telling you something. The data doesn't lie. Only researchers lie. So therefore, the data is telling you something. You can't expurgate it. You have to tell your audience out there, well, this is interesting. Something happened I didn't expect. I have more ZAP70 than I do over here. No one knows why. Obviously, something about the control of the production of ZAP70 was altered in the mutated form versus the wild type form. I don't know why. Okay. All right. That's what I wanted to show you. I saw your hand first. Go ahead.
What was the inhibitor for again? The 5.
I can't pronounce it. It's this. Okay.
Well, what does it inhibit? Like
It
the.
inhibits ZAP70.
Oh, I see. Okay.
Its inhibitor is ZAP70.
Okay.
The wild type is not sensitive to it. Only the
Okay.
mutated form is
Gotcha.
sensitive.
Alright,
So it just inhibits ZAP70's ability to like perform the next step, but it doesn't inhibit the actual activate or like phosphorylation of ZAP70.
That's right, because that's activated by Src kinase.
Right.
Src kinase is not affected by this inhibitor, only ZAP70 is. So everything's fine until after the step where ZAP70 becomes phosphorylated. So ZAP70 is phosphorylated, it should be active, but in this presence of this chemical, which is an inhibitor, it can't carry out its kinase activity. So anything downstream from that no longer is going to occur. And that's what we showed, because LAT was not phosphorylated. Yeah.
What's the vehicle?
Vehicle just means the medium in which we put the cells in, the buffered medium, the liquid medium, that's all it is. That's all it is. Okay, any other questions about that? All right. All right, now, what happens? Now we have two things to talk about. We have to talk about diacylglycerol and IP3, because remember, we've activated phospholipase C, and those are our two products, diacylglycerol and IP3. So what do? Let's start with IP3 because it's the simpler one. IP3 can bind to receptors on the endoplasmic reticulum and lead to the release of calcium. Eventually, the calcium can be drained from the endoplasmic reticulum, and that can open up channels called crack channels, not the crack that is as a pejorative sense, but crack, which is CRAC, which stands for calcium release activated channels. Now, how many people took Bio 46? Now, how many people remember that? From Bio 46? All right, Susan loves this particular channel, so I know she emphasized it. And what happens is the crack channels are calcium channels, which allow extracellular calcium to come into the cell as well. So in this case, you release the calcium, all of it essentially from the ER. When that happens, that activates the crack channel and does so in a way which involves two different proteins, which we're not going to talk about in this class, which communicate with each other in the cell membrane and the endoplasmic reticulum. Suffice it to say for this class that once the ER is empty of calcium, these two proteins communicate and lead to the opening of these crack channels. So more calcium comes from extracellular space into the cytoplasm of our cell. Okay. By the way, these crack channels may also exist in smooth muscle cells. Is anybody in Bio 115 in this class now? probably. So next week we're going to talk about cracked cells and smooth muscles because they exist there as well. They don't really exist in skeletal muscle or in cardiac muscle, but they do exist in smooth muscle. How important they are in smooth muscle is debatable. But anyway, immunology is not the only domain where a lot of this cell biology, these cell biology processes are taking place. All right. So what do we do with the calcium? Well, calcium, which we're also going to talk about next week in smooth muscle physiology in Bio 115, so some things do complement each other. Calcium binds to a regulatory molecule called calmodulin. And I'm sure if you knew anything about basic chemistry, you know that calmodulin has to be a highly negatively charged molecule because it's going to bind avidly calcium. And in one calmodulin molecule can bind 4 calcium ions. That's a lot of positive charge. So you would infer from all this, and correctly, that calmodulin is a pretty highly negative charged molecule. So calcium is avidly seeking, if you will, to be trapped by calmodulin. Once calmodulin binds to calcium, it changes its configuration, and it can bind to a regulatory region on a phosphatase. And the phosphatase that we're concerned with here is called calcineurin. Remember, a phosphatase removes phosphates, the opposite of a kinase. So this molecule of calcineurin is always present in a T cell. However, it's inactive. It only becomes active when calmodulin, after it's bound to calcium, binds to the regulatory site of calcineurin. And now calcineurin becomes an active phosphatase. Now remember, where did we get our calcium? We got it because IP3 bound to the ER, releasing calcium, and once the ER is spent from the ER, that causes increased release of calcium through the channels, called crack channels of the cell membrane. Lots of calcium. All right, so now what does calcineurin do? Calcineurin dephosphorylates a transcription factor called nuclear factor of activated T cells, NFAT. Nuclear factor of activated T cells. This is one of the transcription factors. I said one of the 4 I said we would discuss. In the first slide I showed you today. NFAT is normally phosphorylated and it's inactive. Inactive because it's stuck in the cytoplasm. It can't get through the nuclear pore into the nucleus. But once you activate calcineurin, a phosphatase, then NFAT can go through the nuclear membrane, get into the chromosomal material, and lead to active transcription of whatever genes it's controlling. Probably some cytokine. Now, calcineurin is interesting because this is one of the molecules that in organ transplantation, there's a drug which can inhibit calcineurin. And that drug is given to patients who receive kidney transplants when there's little or no match. And what that drug does is inhibit calcineurin. And when you do that, you can't activate NFAT. And therefore, you can't activate a T cell very proficiently. And therefore, your immune response is suppressed. And therefore, there'll be less rejection of that foreign organ. So this is an important molecule clinically, again, with organ transplantation. This is what is inhibited. There's more than one type out there of molecule which can inhibit calcineurin, and the newest one is less toxic than the older one. So, at any rate. Okay, so that's our first mission. We have 3 more transcription factors to worry about. All right. The other molecule is diacylglycerol. Diacylglycerol does 2 things, one of which is quite confusing, or not confusing, it's quite elaborate, and one which is pretty straightforward. So let's start with the straightforward outcome of diacylglycerol. Diacylglycerol, once it's produced, shown by this, this crooked thing here, is diacylglycerol. Okay. And that remains in the cell membrane as you would expect because diacylglycerol is just a glycerol molecule with 2 fatty acids attached. So it's not going anywhere. It's staying in the cell membrane. And the diacylglycerol can activate a molecule which is now juxtaposed to it because of all this cross-linking, called protein kinase C. Protein kinase A, you know, of course, is activated by cyclic AMP. This is protein kinase C, which is not activated by cyclic AMP, but instead activated by diacylglycerol. Okay, and those people in physiology are going to be talking about this because this is also involving smooth muscle contraction. All right, so now we have protein kinase C. Now, what does protein kinase C do? Well, a number of things, but we're not going to learn all the steps. And what happens is once protein kinase C is on the scene, it activates a whole series of molecules, which we're not going to talk about. You can go home and read these if you want. And on your exam, 50% of your next exam is going to be a blank slide like this, and you have to fill in all the names. I'm not kidding. But at any rate, what happens is diacylglycerol leads to the activation of a whole bunch of molecules, and eventually activates a molecule called inhibitory, IKK. Sounds like we're talking about sororities and fraternities again. This is not iota kappa kappa. This is inhibitory kappa kinase. Okay? An inhibitory kappa kinase binds to a molecule called inhibitory kappa B. Now, our second transcription factor is called remember I told you about this in the very first slide, is nuclear factor kappa B. Remember, there's NFAT we just described, which was activated because of calcium. NF kappa B normally is inactive, sitting there in the cytoplasm because it has this inhibitory molecule on it called inhibitory kappa B. And because of diacylglycerol and protein kinase C, to make a long story short, we're skipping all these steps, is we activate an enzyme called inhibitory kappa kinase. An inhibitory kappa kinase, by its very name, phosphorylates inhibitory kappa B. So now we have diacylglycerol. I know I'm repeating myself, but there's a purpose to my madness, a method to my madness. Diacylglycerol activates protein kinase C, which leads to the activation of inhibitory kappa kinase, which then phosphorylates inhibitory kappa B. Now, inhibitory kappa B is normally stuck onto nuclear factor kappa B and prevents nuclear factor kappa B from translocated into the nucleus. But if you phosphorylate inhibitory kappa B, inhibitory kappa B falls apart or moves away, moves out of this pocket in nuclear factor kappa B. And once inhibitory kappa B is gone, nuclear factor kappa B migrates into the nucleus, acting as a transcription factor, and now you transcribe additional genes. Now, I want you to digest these two things. I want you to digest calcium induced by IP3, and then I want you to digest protein kinase C activated by diacylglycerol. So anyway, now digest it, put an engram in your head, All right, let's do it as a class then. So we activated phospholipase C, we get what? Get IP3 and diacylglycerol. IP3 what? Binds to the ER, you get releases calcium, the calcium then releases more calcium from the crack channels in the cell membrane, you get more calcium. Calcium binds to calmodulin, a highly negative molecule binds to calcineurin. Calcineurin is a phosphatase or kinase?
Phosphatase.
And it dephosphorylates what?
NFAT.
NFAT, and it dephosphorylates, and now and only then NFAT can go into the nucleus into transcription. Okay, we're a quarter of the way done. Next is diacylglycerol. Diacylglycerol does what? It activates which important kinase? Protein kinase C. Not A, not B, not G, heaven's sake, there's a G, which I'll also talk about in the muscle physiology week. But you have protein kinase C, not directly, there's intermediate steps which are skipping, inhibitory kappa kinase, which then phosphorylates what? Inhibitory kappa B, which is stuck onto nuclear factor kappa B, and when inhibitory kappa B becomes phosphorylated, it disassociates from nuclear factor kappa B, which then migrates into the nucleus, leading to active transcription. And now we're half done. Okay, so there you go. All right. Wow, this is really fascinating, I think. All right. Okay. Next role of diacylglycerol is to activate a signaling pathway for the rest of your lives. You have to know about. And I'm not kidding you. For the rest of your lives, this is a very important signaling cascade, no matter where you go in the world after this. You will have to know the MAP kinase cascade. The MAP kinase cascade, no matter where you go in life. And what does MAP stand for? Mitogen, activated, protein, kinase cascade. Remember, I'm 70 years old. If my mind can remember all this crap, your young minds are much more agile than mine. You should be able to integrate this into, again, your neuronal pathways really, really easily. All right, so now we have our next step is to activate the MAP kinase cascade. That's also done by diacylglycerol. But before we learn that, we have to know some certain names. And this may be repeated. How many people know about the MAP kinase cascade? Keep your hands up. All right, how many people know about the MAP? Be proud. All right, how many people know the MAP kinase cascade? Those people with your hands up, I won't pick on you. Tell me what it is. Can someone tell me who knows what it is? Tell me what it is. Yes, thank you.
Pathway that leads to proliferation.
Sorry?
It's a pathway that leads to proliferation.
Can, right? It does. It's mitogen activated, so it means it's involved in mitosis, absolutely correct. But it's involved in many, many other signaling processes as well. Does anyone know the steps involved? How many people took Bio 46? I know, Susie. I know she spent a lot of time on this. All right. So, all right. Let's review it. MAP kinase cascade involves a bunch of characters that we have to know all their names. One is something called a small G protein. Here's our small G protein. And that's what they're called, small G proteins. Small G proteins are monomers. Most G proteins we're familiar with in cell signaling are trimers, heterotrimers, with an alpha, a beta, and a gamma subunit. But that's not what we're talking about here. We're talking about, in the MAP kinase cascade, small G proteins, which are always monomers. And a G protein is so called because G proteins combine guanyl diphosphate. That's what this is. Guanyl diphosphate. That's why it's called a G protein. They combine avidly, not ADP, but GDP. Hence the term G protein. And a G protein doesn't do us any good unless it binds GTP. The inactive state of a G protein is one that has GDP in its binding cleft. So to get a G protein activated, you have to rip out that ADP and exchange it for GTP. That's pretty easy to see here, the triphosphate. And here's another cast member. The molecules which can rip out the GDP and exchange it for GTP, here's the DP, 2, or TP, here's the 3, is guanyl nucleotide, G, exchange factor, GEF. GEF exchanges one type of guanyl nucleotide for another. GEF rips out a diphosphate and substitutes it for a triphosphate. So we rip out the GDP, the GEF, ripped, and now we have GTP. And then this small G protein can be involved in cell signaling. How do we turn it off? Turns out this small G protein also has GTP-ACE activity, and therefore it's in the on state for a brief period of time. because the G protein, because of its GTPase activity, can split GTP back to GDP, and then we're in the inactive situation again. So you have to know these cast of characters' names, otherwise I'm going to lose you. So we have, first of all, small G protein, which is always a monomer. G protein because it binds a G, DP or GTP. To get the molecule in the active state, we have to rip out the DP and put a TP in there. And to do that, we need a guanylyl nucleotide exchange factor. And that's what is called a GEF. And the GEF puts a TP for the DP. And this state, the on state, only exists as long as the GTPase doesn't convert the TP back to the DP. And then, by the way, there are other molecules called GAPs. Not where you buy clothes at, but the GAPs are called guanylyl nucleotide accelerating factors. They speed up the GTPase of the G protein, so we can have controls that way as well, but we're not going to talk about GAPs in this class. I thought I'd throw it in there. Okay, so what does this have to do with diacylglycerol? We'll learn that in a minute. We'll learn that in a minute what this has to do with diacylglycerol. But here's the MAP kinase cascade. Here's the classic MAP kinase cascade. First of all, we have a GEF, or guanylyl nucleotide exchange factor. I said you have to know these cast of characters' names. Here's our small G protein, which will now have a GTP. Once it gets GTP, it's an active small G protein. And the small G protein, by the way, when it's active, does not have an enzymatic effect. It's not an enzyme, other than having GTPase activity. It's not a kinase or a phosphatase. So I'm going to disabuse you of that idea. It's not a kinase or a phosphatase. And the target for the active small G protein is a MAP kinase. And the reason MAP kinase is called kinase because the MAP kinase phosphorylates at one site an enzyme is called a MAP kinase. So the MAP kinase, I like this every year when I teach it because everyone gets this look of either despair, disgust, or amusement. So the kinase phosphorylates at one site, either serine or threonine, on an enzyme which is called MAP kinase, which then in turn phosphorylates at two sites, a MAP kinase. So MAP kinase is called MAP kinase because it phosphorylates its target at two different amino acids, hence why it's called a kinase. Kinase is called that because it phosphorylates a kinase. Get it? So it's a kinase of a kinase, hence MAP kinase, which then phosphorylates a MAP kinase, which has a target of two amino acids, okay, a tyrosine and threonine. And MAP kinase then phosphorylates the third character here, the MAP kinase, which should have two phosphorylations, but it's only showing one here. It should have two phosphorylations. Should throw another one in there. Okay. So, all right. So the names of the kinase, the kinase, and the kinase we're concerned with, and these aren't always the names, are RAF, MEK, and ERK. You have to memorize those. RAF in that order. MEK and ERK. RAF, MEK, and ERK. RAF, MEK, ERK. Okay. Sounds like someone's name. Ralph, Michael, ERK. All right. Think of it that way. Ralph, Michael, ERK. I guess that's a good way of memorizing it. Ralph, Michael, ERK. So Ralph, Michael, and ERK. Okay. And RAF is the kinase. What is the kinase phosphorylate in three places? No, just one, right? But then it phosphorylates a kinase, which does phosphorylate in two places. By the way, ERK should have two phosphates on this cartoon. I just realized is incorrect. It should have another phosphate. All right, so our next step is to figure out how diacylglycerol is involved in activating this MAP kinase. Now, I typed up here, actually, when RAS has a GTP associated with it, it dimerizes. Remember, that's usually the rule of thumb. Things to become active dimerize. And when RAS dimerizes, it then leads to the dimerization of RAF. And then RAF undergoes a very complex biochemical change, becomes partially dephosphorylated and then phosphorylated at another site. So it undergoes both dephosphorylation and phosphorylation, and also proteolysis. So under a very complex series of steps take place, which we're not going to talk about in this class, which I'm sure Dr. Cagle covers in Bio 46. I assume she would, but we're going to skip that in this course. Okay. In any case, I typed in probably more information than we need to know. But I want you to, well, I do want you to know is this molecule, RAS, is not either a kinase or a phosphatase. The only enzymatic activity the small g protein has is GTPase activity. It doesn't have any other enzymatic activity. So we typed in the relevance, but the thing here is you get dimerization and then activation. Did they cover this in biochemistry? They talk about how RAS dimerizes and becomes active, and they talk about that. Okay, so maybe you want to tell us that? Okay, which biochemistry did you take? 152 or 171? Okay. And we take 152. Did you do this in 152?
No.
Yes?
No.
No? Who cares, right? Now we have diacylglycerol. Diacylglycerol. What is diacylglycerol? One thing it does is it binds protein kinase C to the activation of protein kinase C. But in addition, it also binds RAS grip. RAS grip. Which I pronounce it grip. I just like to say that. GRP is the GEF for RAS. And remember what GEF stands for? Guanine nucleotide exchange factor. So this molecule, okay, RAS grip, drags out the GGP from RAS and substitutes instead of GGP, GTP, and now RAS is active. And RAS, by the way, doesn't have kinase or phosphatase activity. No, it only has GTPase enzymatic activity, but it causes dimerization of RAF. Remember our RAF, right? Okay, that's important to keep in mind. All right, so now we have RAS grip. RAS grip leads eventually to our activation of our MAP kinase, then a MAP kinase, and then finally MAP kinase. And our MAP kinase, in this case, is ERK. Remember? Ralph Michael ERK? So ERK is the third MAP kinase here. And you notice this is correct. In this case, it's doubly phosphorylated. It has two phosphates on it. Because remember, the kinase leads to phosphorylation of two sites. And then ERK translocates into the nucleus where, being a nice kinase, it phosphorylates something called ELK. So there's an animal at the end of this whole thing, an elk. An elk leads, as a transcription factor, leads to transcription of another transcription factor. So it's a transcription factor leading to transcription of another transcription factor called C-Phos. And now we've done 3 of the molecules I promised to deliver today. We've done NFAT, nuclear factor kappa B, and now C-Phos. Now how do we get C-Phos activated? Let's go through that again. We got diacylglycerol. Diacylglycerol, let's leave aside protein kinase C, also binds and activates RAS grip. And RAS grip is what type of molecule? A GEF, which drags out GDP and substitutes for GTP on our small g protein called RAS. And is RAS kinase or a phosphatase? No. But it causes dimerization of RAF, and then a whole series of biochemical steps which we get active RAF, which then leads, RAF actually gets phosphorylated as well. You see a little lollipop on it. And then RAF activates MEK, which is a protein, which is a kinase, which then phosphorylates ERK, which is doubly phosphorylated, which then translates it to the nucleus, which then activates ELK, and ELK is the transcription factor then, which gives us C-phos. So the last step of all is to get C-Jun. That's the fourth thing I promised to teach you today. C-Jun. So we have C-phos. Where the heck does C-Jun come about? Comes down on the bottom here. This is another MAP kinase cascade. I typed out a whole bunch of stuff of names you don't have to know. But this second MAP kinase is probably activated. What's the name of the co-receptor? TCR is the primary receptor. What's the co-receptor called? CD28, which binds to what on our APC? CD86 or 86. And when CD28 binds to CD80 or 86, that activates another MAP kinase cascade, and the end point is to C-Jun is in the cytoplasm, and then translocates into the nucleus. And it turns out C-phos dimerizes with C-Jun to form our ultimate transcription factor called AP1. Now, we've done all 4 transcription factors. I've done NFAT, nuclear factor kappa B, C-phos, and C-Jun, which is the dimer, gives us AP1. All right, so what does diacylglycerol do? Diacylglycerol activates protein kinase C, which essentially inhibits inhibitory kappa B, which then gives you nuclear factor kappa B, which then translates to the nucleus. But then you also get activation by diacylglycerol of RAS GRIP, and RAS GRIP then gives you activation of RAS, which then gives you activation of RAS, which gives you activation of MEK, which gives you activation of ERK, which gives you activation of ELK, which gives you production of C-phos. All right, and then how do we get C-Jun? So we completely other MAP kinase cascade, probably activated by CD28 binding to its ligand, which is CD86, and then we get C-Jun, and C-Jun, once it's phosphorylated, can translate to nuclear C-phos, and we get C-phos C-Jun, which gives us a master transcription switch called what? AP1, AP1. Okay, brilliant. Now, if I inhibit ZAP70, making a long story short, will I get ERK activated? Will I get ERK phosphorylated? Look. Will I get ERK phosphorylated? No. Okay, so ZAP70 is necessary to become activated in order to get ERK phosphorylated, that is. And if if ZAP70 is not activated, then we won't get ERK being phosphorylated. Okay. Susan's research has really taught us a lot there. Okay, now look at this slide and ponder it for 2.5 minutes. This is most, not everything. There's some stuff left out here, but most things I told you about today are on here. So look at it. Now, do your homework now. Look at it. CD45 isoforms, I tell you about that because I'll delay that. There's different types of CD45s in T cells, which we'll learn about today. So forget about the CD45 isoforms today. Naive and memory T cells have different forms of CD45. We can distinguish memory cells from naive T cells by the type of CD45 they have. But we're not going to do that today, even though I'm blabbing to you anyway. Yeah.
Is RAS GRP G protein?
RAS GRP is not a G protein. It's a guanylucleotide exchange factor. RAS is the G protein.
So you said RAS and other G proteins don't have enzymatic activity, but can cleave?
No RAS small g protein has kinase or phosphatase activity. They have GTPase activity, so the only thing they can do. But when they bind to RAF, they cause dimerization of RAF, and then RAF, remember, there goes all these biochemical changes as a result, one of which ends up being phosphorylated on the T site.
But why?
But not by RAS.
Okay. But why does it need catalytic activity if GEP can just substitute GDP for GTP?
Because the RAS is not active unless it has GTP at that site. GTP, the molecule, won't cause dimerization of RAF.
Oh, okay.
Okay? So in order to get dimerization of RAF, you have to have GTP stuck into the RAS grip.
So that doesn't need like any catalysis that you mentioned from GTP, from the G protein, because you said
No, well,
it has,
the
like,
G
cat-
protein, I said the only enzymatic activity it has is GTPase activity.
Oh,
It doesn't have
GTPase.
kinase, it doesn't have phosphatase
Oh,
activity. It's the only enzymatic activity it has. Is GTPase activity, not kinase or phosphatase. So RAS does not phosphorylate RAF directly. It causes dimerization of RAF, which then leads to its phosphorylation. All right, everybody review this. Let's do it together as a class real quickly. Okay, let's have some fun here and see how fast we can go. All right, mistake number one. We're only showing what? One what? And we all know by now, in order for this whole process to work, you have to get what? Cross-linking. And then this cross-linking brings our whole mess into what part of the cell membrane? A lipid? Raft. Okay, by the way, rafting is fun, but not in the winter. All right. So then, once you get this all clustered in the middle, CD45 dephosphorylates which important enzymes? Sarc kinases, which then leads to the phosphorylation of what? ITAMs on our zeta, which then act as a docking site for what? ZAP70. And what does ZAP70 end up doing? Phosphorylated. And when ZAP70 is phosphorylated, it becomes an active what? Tyrosine kinase. Okay, and tyrosine kinase, once it's phosphorylated, the ZAP70 is phosphorylated. Once it then phosphorylate, Latin slip, which forms a basket, which then forms the binding site for phospholipase C, which then gets phosphorylated by a tech kinase, which then phospholipase C, once it's activated because it's phosphorylated by a tech kinase, what's the target for phospholipase C? Phosphatidyl inositol. And what are the two products of that attack? Diacylglycerol and IP3. IP3 binds to the ER, leading to release of calcium, which leads to more calcium from the what type of channels? Crack channels, okay. More calcium comes in. Calcium binds to calmodulin. Calmodulin binds to calcinurin, which is a phosphatase which dephosphorylates NF-AT, which then translates to the nucleus to give us active transcription. Okay. All right. Now, nuclear factor kappa B has what? Inhibitory kappa B associated with it. Okay. Diacylglycerol leads to the activation of what? Inhibitory kappa kinase. Inhibitory kappa kinase in turn then leads to the activation of nuclear factor. Come on, look at how nuclear factor kappa B, which also gives us transcription. All right, the last thing of all, diacylglycerol activates RAS grip. RAS grip is a GEF then exchange. This is GTP or GDP on RAS, and RAS then activates RAF because of the dimerization, and then it becomes a phosphorylated, then it phosphorylates what? RAF comes right after that MEC, and MEC becomes what? Our kinase, so then it phosphorylates ERK, which becomes doubly phosphorylated, and ERK moves into the cell nucleus, and then it phosphorylates L, which then gives us what? C. Phos, and then how do we get C-June? Through the CD28 pathway. When we get the CD28 pathway, finally, class, one more minute. What do you get? AP1. Okay. And there, you got your 4 transcription factors I promised you, and now you're off and running and understanding T-cell signaling. We still have a few slides to go through. We'll continue this on Tuesday. By the way, if you got lost in that last explanation, everything is on this slide. Everything. So this is your homework. Learn this slide.